Hi, everybody. Welcome to Chapter 10. This is the first of the many lessons that we'll be covering this chapter, which the major topics are going to be the two types of cell division, mitosis and meiosis. Before we jump into those topics, though, I just want to do a little introduction to kind of some cell characteristics, and then we'll jump into those processes. So this first lesson is just some general information about the human genome and the cell cycle. So the human genome, when we talk about that, is just the collection of all the genetic material inside of your cells. So for humans, our human genome consists of 46 individual chromosomes, 23 pairs. So what that means is you have one chromosome, number 1 from mom, and one chromosome, number 1 from dad. So you have two number 1 chromosomes. When you were fused, when you became a being, chromosome number 1 came in mom's egg, chromosome number 1 came in dad's egg, fused, and now you have 2 of each of those chromosomes. So then you will have chromosome number 2 from dad and chromosome number 2 from mom. So you have 2 each of the chromosomes. And then for the sex of the individual, we have either XY is a male, oops, let's get a darker color, is a male sex, and XX is a female. Since females only can do X's, the X comes from mom or dad. The Y can only come from dad. So it's really the sex of the baby is determined by the dad's sperm. So the dad will produce about half of all of the sperm will be carrying X's and about half will be carrying Y's. All of mom's eggs will be carrying X's. So depending on whatever combination comes together, that gives you the sex of the baby. So that's our human genome. And you can see they're usually typically, like in a picture, they'd be ordered from the biggest. Chromosome number 1 is the biggest, chromosome number 2 or 22 is the smallest, and we can see there's quite a bit of difference between the X and the Y chromosomes. I'll get rid of all that writing. X chromosomes are relatively big. They hold a lot of genes, more than just the genes needed for the femaleness. Y chromosomes are pretty small. They really only carry genes for maleness as far as the sex goes. All right, so that is our human genome. When we take a look at where we find this, so if we go back to our knowledge about cell structure and function, we remember that our DNA is housed within our nucleus when we are talking about eukaryotic cells. So here is our DNA, we might recognize this double helix made up of nucleotides in that the repeating are the A's, T's, C's, and G's. So it is not just hanging out in our cells as just as loosely like a ribbon of DNA. It really is more organized and packed a little bit more efficiently. So the way that the DNA is packed or packaged inside of your cells is the DNA molecule itself is wrapped around these proteins called histones. So kind of like this. Wrapping around. So these are called histones. They're found in eukaryotic cells. Prokaryotic cells do not have these. And then the histones kind of package in themselves, now we have some histones folding and folding and folding. And this is what we call a 30 nanometer chromatin fiber. So when we are talking about a cell that is not in the process of getting ready to divide, it's just a regular old cell being a cell, it is at this stage that most of your DNA is just kind of hanging out in what we call chromatin. So it's wrapped around, the DNA molecule is actually wrapped around the histones, and the histones are kind of folded in on themselves, and this is what we call the 30 nanometer chromatin. In preparation for division, though, when we start getting into the steps of what happens during mitosis and meiosis, when we say the chromosomes are condensing or supercoiling, then they start going into these loops, and the loops start looping, and the loops start looping more and more and more and more. So what you end up with is a super coiled, tightly bound packet of DNA into what we typically see as often you see a chromosome kind of like this. So that's just super coiled, and it's only in this form in the processes of meiosis and mitosis. it's not staying that way. So once it's done with the whole cell division, it will go back and kind of hang out in this phase. So the chromatin is the loosely coiled, it's not the straight out just loose DNA, but this is kind of the loosely coiled is our 30 nanometer chromatin fiber. If we were to compare our genome with other organisms, we can see there's some variety. Not all organisms have a chromosome number of 46 or, a pair of 23. So we can kind of see here's human down at the bottom. Oh, let me get a different color. So here's human down at the bottom. We have 23 chromosomes in haploid. We don't really know what haploid means. Haploid just means half the number of like one set. So we have 23 individual chromosomes. In most of our cells, we have 46 total, which is a pair of each of the 23, with about 3,000 what's called megabases. So megabases is just a unit for measuring how many A's, T's, C's, and G's. So each one of those is a base. So comparing us to other organisms, say like another mammal, like a cow, we have about the same size of genome, about the same number of bases in our chromosomes. But if you take a look, they actually have more chromosomes than we do. So their haploid number is 30, which means their diploid number, their complement of 2 sets, they'd have 60 chromosomes, where we only have 46. Now, let's compare something else, all right? Let's take a look at a chicken. It's a bird. Look at its genes. It has, or not genes, but its size of its genome, 50,000 megabases on 39 chromosomes. Now, I display this just so you can get the feeling or the understanding that complexity or intelligence or adaptability is not dictated by the number of chromosomes or the size of the genome. Clearly, we are more advanced than chickens and cows, but they have more chromosomes or more actual bases than we do in our genome. So again, there's a wide variety, like a lily, a plant, has the same amount of DNA that a cow does, or a chicken, sorry, the chicken. That doesn't mean that the lily is more complex than the animal of a chicken. It just means that's its genome. Every organism has its own unique amount of DNA in the number of, and a varying amount of number of chromosomes. Okay, so just kind of some cool factoids there for you. So the last slide in just this introduction is to get you comfortable with the terminology of the cell cycle, because we're going to spend a lot of our time here in the M phase. That's mitosis, which is pretty much the half of chapter 10. But we do need to know what's happening in the entire cell cycle. So a cell cycle you can think of as the lifespan of a particular cell, and it is found in these different phases. There's 2 major divisions of the cell cycle. We have what's called the M phase, and we have what's called interphase. So the M phase is a cell that's actively dividing. It's going through all the prophase, metaphase, anaphase, telophase, cytokinesis. It's all doing that division. But then the rest of the time of the cell's lifespan is spent in this interphase, sometimes called the resting phase of a cell's lifetime or lifespan. So interphase is broken up into 3 steps, and they're numbered G1, S, and G2. The G stands for gap. So it's the gap phase. Sometimes I like to call it the growth phase because that's really what's happening. So there's lots of growth. The cell is doing its thing. So it's a liver cell, it's being a liver cell. A skin cell, it's being a skin cell. Whatever, intestinal cell, it's being an intestinal cell. Pancreas, it's doing what it needs to do. This G1 is kind of just like I'm hanging out, I'm being a cell. If a signal comes along and says, hey cell, it's time for you to divide, so maybe like a skin cell, we have to replenish the ones that are lost on the surface, it will get some signals and it will move into the S phase. The S phase stands for synthesis, but specifically DNA synthesis. This is when DNA gets copied. Okay, so the S phase of interphase only happens before a cell is getting ready to actually divide to go into the M phase. The second gap, or gap 2, is another little growth stage. It's a lot shorter than the G1. This is when the cell is in preparation for mitosis or meiosis. It's prepping. It's making copies of its organelles. It's growing a little bit bigger. Maybe it's making more endoplasmic reticulum or mitochondria or more nuclear envelope because we know it's getting ready to become 2 cells shortly. And then the signals will come in. G2 will then move on into the M phase, and then that 1 cell will become 2. So that's what we're going to spend the next video on, will be mitosis, which again is going to focus on those stages of cell division. Yeah, so that's it. So that's just a quick little introductory video. I will see you next time when we run through the steps of mitosis. Okay, bye.